Some transplants do not succeed. Interestingly, transplantation follows principles similar to human relationships. Compatibility determines acceptance or rejection. To understand this, let us begin with the types of transplants. First, we have the autograft. The best example is skin grafting, where a piece of skin is taken from one part of the body and grafted to the damaged area in the same person. Next, we have what is called as an isograft. Here, the tissue is transferred between monozygotic twins. This is followed by the most commonly done method, what is called as an allograft, where the tissue is transferred between genetically different individuals of the same species. Finally, we have the xenograft, where the tissue is transferred between members of different species. What are the graft? The acceptance is mainly dependent on the concept of histocompatibility. Histocompatibility means between the graft and the recipient. This decides whether the graft is going to be accepted or rejected. If the graft and recipient tissues are compatible, then the graft is accepted. So generally, autografts and isografts are compatible. Histo-incompatible grafts are generally rejected by the recipient. So some allografts as well as xenograft may usually be histo-incompatible. The transplantation antigens are the antigens of the graft against which a recipient can mount an immune response. The major player in this is the MHC, or the major histocompatibility antigens, which is screened by the recipient's immune system very keenly. Another factor is the blood grouping and RH typing between the graft and the donor and the recipient. We also have what is called as a minor histocompatibility antigens. These are some peptides derived from normal cellular proteins of donated organs. The immune response against these molecules is weaker, so there is a lesser chance of rejection. One exception is when a graft is transferred between a male donor to a female recipient. This phenomena is called as a Equal-Slimmers effect. The graft tissue of the male donor would have some male-specific minor histocompatibility antigens, which is determined by the Y chromosome, which is absent in the female recipient. So it has been observed that the rejection of grafts when transferred from a male donor to female recipient is more compared when it is vice versa. Now let us see what are the types of rejection clinically. We have the hyperacute rejection, acute rejection, and the chronic rejection. First, let's talk about hyperacute rejection, which happens within minutes to hours after transplantation. Why this happens? It is mainly because of the thrombosis of the graft vessels and ischemic necrosis of the graft, which is because of the circulating antibodies in the graft cells. Exposure to foreign HLA antigens can occur maybe because the patient has been transfused early, patient has been pregnant where she has been exposed to foreign antigens, or previous organ transplantation. This leads to preformed antibodies which will act against the graft. If an individual with a pre-existing antibodies receives a graft containing the same foreign HLA antigen, the graft will be rejected earlier and more vigorously. Generally, this does not happen because we have a lot of procedures before a transplantation in the form of screening. Next, we have what is called as acute rejection occurring within days or weeks post transplantation. It is due to active immune response of the host stimulated by the alloantigens in the graft. Acute rejection is mediated by cytotoxic T cells and antibodies specific for the antigens in the graft tissue. The cytotoxic T cells destroy the graft cells. Helper T cells, on their part, will secrete cytokines which will cause inflammation in the graft. Antibodies will ensure the graft blood vessels, which is mediated by the classical complement pathway. Current immunosuppressive therapy is designed mainly to prevent and reduce acute rejection by blocking the activation of these reactive T cells. Finally comes the chronic rejection happening over months or years post transplant. Here, what happens? There's a fibrosis of the graft and gradual narrowing of the graft blood vessels, which is called as a graft atherosclerosis. T cells that react against the graft antigens secrete cytokines. This will stimulate the proliferation and activation of fibroblasts and the vascular smooth muscle cells in the graft. This will lead to a form of chronic delayed hypersensitivity reaction. The alloantibodies also contribute to chronic rejection, and this is one of the main reasons for a higher number of graft failure in transplant recipients. Now, let us look at the factors responsible for this rejection. Some grafts are rejected faster than others. Example is the skin compared to either kidney or heart. It is rejected much more faster. The genetic distance between the donor and recipient also plays a role. So autografts and isografts are well accepted. And immunological memory rejection is faster when there is another graft is placed onto the recipient from the same donor, and this occurs because there's a memory cells produced against the first graft would definitely and quickly differentiate into effector cells which act in rejection of the second graft faster. Now let us see what is the normal graft acceptance process. The graft begins to revascularize in 3 to 7 days, and the healing process starts around 7 to 10 days, and the graft is fully accepted by the end of 2 weeks. So, what will happen if the graft is not compatible with the host? Once the revascularization phase starts by 3 days, the immune response also starts. By 10 days, there is a decreased blood supply of the graft, and the complete rejection happens by the end of 2 weeks. So, this is called as a first set rejection response. If at all we try the same graft again from the same donor, the rejection process will happen in a much more faster fashion manner, and the graft undergoes necrosis by the end of first week itself. So this is called as a second set rejection response. Graft rejection basically is a T cell mediated immune response to the graft antigens and MHC molecules. The graft brings in new MHC molecules. The recipient's immune system recognizes the new MHC molecules. The graft peptides are presented to the T cells. This leads to sensitization, and the final outcome is immune mediated graft rejection. The sensitization can happen either via a direct pathway or indirect pathway. In the direct pathway, the antigen presenting cells with the MHC is presented to recipient immune system. This leads to graft rejection by cytotoxic T cells. In the indirect pathway, the graft cells are ingested by the recipient APC, and they are processed. They are presented to the MHC molecules of the host and to the helper T cells. This pathway is responsible for most of the chronic rejection mediated by helper T cells via specialized form of a chronic delayed hypersensitivity reaction. This leads to effector phase, which can be delayed hypersensitivity type or action of cytotoxic T cells. There's another method which is the antibody mediated cells, which can be complement mediated lysis or antibody dependent cell mediated cytotoxicity via the natural killer cells or macrophage mediated destruction. Now, to assess the compatibility, the major component is HLA typing and the ABO matching. So, first let us see about the HLA typing. In this test, the donor's antigens expressed on the surface of leukocytes or their gene of that recipients are matched. The HLA compatibility, you have two methods of testing. Either it can be a phenotypic, which includes serology like microcytotoxicity, tissue typing, which is called as a mixed lymphocyte reaction. Genotypic methods, you have the PCR detecting HLA genes, PCR restricted fragment length polymorphism, PCR sequence specific oligonucleotide probing, PCR sequence specific primers, PCR DNA sequencing, and conformational analysis. Among this, the PCR specific oligonucleotide probing, the PCR sequence specific primer, and PCR DNA sequencing are more reliable and which are currently in use. Obviously, you have the ABO blood grouping compatibility testing for blood grouping and cross matching. Once the transplantation is completed, hyperacute rejection can manifest severely in within minutes. So the treatment indicated is immediate removal of this tissue. Acute rejection, on the other hand, you can try some regimens, therapeutic regimens, or a combination of various immunosuppressive agents. So these immunosuppressive agents, you have various varieties. You have the corticosteroids, calcineurin inhibitors, mitotic inhibitors, antiproliferatives, and mammalian target of rapamycin. So you have this prednisolone, cyclosporin, azithromycin, cyclophosphamide, all those things. The next most important type is a monoclonal antibody, which the antibodies are based against the CD2 molecules or CD3 or CD4 on the T cell surface, anti IL2 or alpha receptor antibodies, monoclonal antibodies to C20 antibodies, monoclonal antibody to tumor necrosis factor alpha, you have the anti thymocyte globulin and anti lymphocyte globulin. Now, the chronic rejection is generally considered irreversible and it is not very responsive to treatment. So, retransplant is generally indicated if possible. Cyclosporin is being investigated for delay or to prevent the chronic rejection of lung transplants. One of the most important questions in this topic is regarding graft versus host reaction. So, generally, what happens? The host will mount an immune response against the graft. This is the opposite. The graft mounts an immune response against the host and rejects the host. This GVH reactions to happen, you have three main conditions what is required. That is, the graft must contain the immunocompetent T cells. It may be bone marrow, thymus transplant, or stem cells. The recipient should possess transplantation antigens that are absent in the graft. The recipient must be immunologically suppressed and therefore cannot mount an immune response against the graft. So, this GVH reaction you can classify into two types. You have the acute or fulminant graft versus host reaction disease, which occurs within the first 100 days of post transplantation. It's a major challenge in case of a bone marrow transplant. The chronic GVH occurs in a less severe form, and anything that is happening after 100 days of transplant comes under this category. Clinically, the acute reactions is characterized by selective damage to the liver in the form of hepatomegaly, skin rash, mucosal damage, and intestinal effects like diarrhea, which is mediated by the graft's immunocompetent T cells. Experimentally, this reactions can be produced in mice, which is called as a Runts disease. Chronic GVH will also attack the above organs, but in addition, it can cause damage to connective tissue and exocrine glands. The treatment is glucocorticoids administered IV, which is given for both acute as well as chronic GVH. So, to conclude, for a successful transplantation, understanding of immunity is important. I hope I have presented this topic in a simple and easily understandable manner. Please do like and share this video and subscribe to my channel. Thank you. Thank you for watching.